Okay, so here we're looking at Self-Growth Division Chapter 10. The cells are limited because our bodies are lazy. They can only get to be a certain size. If they get too big, then the nutrients cannot make it all the way on the inside. The waste cannot make it out in a quick enough time for the cell to be able to carry out all of its processes. So we are limited by the passive transport mechanisms as well as the surface area to volume ratio that is there. Then what will happen is once a cell becomes bigger, then it will need to divide, or it may be able to, or it will reach a certain, mature size, and then it will be able to function then. But organisms that have a need to be bigger will need to be multicellular organisms. So here we have the cell cycle that is basically the life cycle of a cell. Okay, the life cycle that we have. So in any point of the cell cycle, you can start at any point and you can end at any point. It just keeps going round and round and round. All right, so if we look to see, we have the M, okay, this stands for mitosis. This is when the cell nuclear contents will divide. Okay, the nuclear contents will divide. Okay. Once that happens, after these steps, prophase, metaphase, anaphase, telophase, once those steps occur, then there's cytokinesis, and the cell will either go on to grow a little bit and maybe make some more, this G1 equals growth 1. And then the cell basically gets bigger, and it will make some more organelles, makes more organelles, gets bigger. So some growth and some preparation then for DNA synthesis. Once it has grown and reached a certain size, then we have the S phase, and the S phase is DNA replication. And that's when all of the chromosomes within that cell are going to make a copy of each other. So S stands for synthesis, DNA synthesis. Synthesis is making, so DNA replication. I'm not sure why they didn't call it R, but they called it S. Okay. Then the cell will go into G2. So G2 is growth 2. And here the cell will get ready for mitosis. So in order for one cell to become 2 cells, it needs a lot more organelles. It needs maybe a little bit more cytoplasm. So all that's going to be happening in growth 2. So very similar to what's happening in growth 1 is also happening in growth 2. We have DNA synthesis happening in the middle. Then we have the stages of mitosis, which we'll go into more detail in a few minutes. After that, some cells will go into G0. That basically means that they are not going to replicate again. Okay, no more replication. These are cells such as nerve cells. Okay. Once a nerve cell is made, it goes into what we call G0, and it will be there, and that's why nerve damage is so difficult to repair, because the cells do not replicate for nerve tissue. We have not figured out a way to be able to get adult nervous tissue to be able to replicate through. The cell cycle is controlled by cyclones. Okay. These are the chemical signals, cyclones, that decide what stage the cell will be in, how quickly the cell will divide, how quickly it divides, and whether or not it even divides at all. So what happens if there's a problem with the cyclones? What happens to that cell if there's a problem with the cyclones? It can die, or it does not control properly. So what do we call it if a cell keeps dividing and dividing and dividing and dividing? Okay. If it's going to keep dividing and dividing and dividing and dividing. There's a very big disease, begins with a C. Cancer. Okay. Mitosis. Okay. Yes. Yeah. Mitosis equals nuclear division. Division. Okay. So our cell in a normal state is in interphase. Okay, interphase is your basic normal cell. So here we see a picture of a whitefish, an animal cell. Here we see the nucleus. Okay, the nuclear envelope is right here. Here is the cell membrane going around, and this is a drawing. So here we see the cell membrane, then we have the nuclear envelope, which is the nuclear membrane, and we have these two things called centrioles. If you remember, those help with cell reproduction in animal cells. Okay. Our first formal step of cell in mitosis is called prophase. Okay. With prophase, we have pairs of chromosomes visible. Okay, here we see the chromosomes are paired up. This is our nuclear membrane. We see the centrioles are splitting. So in prophase, we have these different pairs of chromosomes here. These are the paired chromosomes, and they are going to be condensing and becoming visible. So they're getting ready to separate. So we have our first one, prophase equals pairs. P, P. Prophase, pairs. Okay, our next phase is metaphase. Okay, in metaphase, what do we notice about the location of our chromosomes? Middle. Metaphase equals middle. The chromosomes are lined up in the middle of our cell, called the equatorial plate. And here we can see in the whitefish cell, the chromosomes are lined up nicely in the middle. So we have pairs prophase, middle for metaphase. Okay, in our next section we have anaphase. What do we see is happening with the paired chromosomes in anaphase? Think of the word A. They are moving apart. Okay? So here we see the chromosome pairs are now separating. They are going towards each of the poles with the spindle fiber, the microtubule, that is helping them to be able to separate and end up in two different locations. So here we see anaphase, these chromosomes moving apart from one another. And our last part is called telophase. What do we see here? Telophase. Telophase.
Twins.
Two. Okay. Here is where we begin to see two distinct cells. Okay, they're not quite two distinct, but this is where we're going to begin to see the two. We can see there's actually a difference here between this side and that side from one another. And here we have the nuclear envelope is going to begin to reform, and we are going to see a few more changes within those chromosomes are going to be on each side, and they're going to start later on to be able to unblind. This last part, which is not part of mitosis, is its own part called cytokinesis. Okay, mitosis is the nuclear division. Cytokinesis is cytoplasm dividing. And it doesn't mean just the cytoplasm, it means the cytoplasm as well as all the organelles. So in cytokinesis, we have all the organelles splitting into the two different sides as well as the cytoplasm. Once cytokinesis is done, we now have two genetically identical cells. We started with one cell and we made two cells. Mitosis is going to help repair tissues. It's going to help us to grow, right? Growing. We need to eat lots of food so we can grow, get bigger. Okay? It enables us, all of our bodies, okay, our entire body is formed by mitosis starting after the moment of fertilization. So once fertilization occurs, our body forms our entire being by the process of mitosis. Okay, the only exceptions to mitosis the exceptions are the formation of the gametes. What are our gametes? Do you know what gametes are? Okay, gametes equal the sperm and the egg. They form by another process called meiosis.